Hey guys, my name is Jonathan. I'm here to present a short discussion on clonal selection theory as it pertains to B lymphocytes for cell biology, the additional oral presentation. So we all know that antigens are macromolecules which provoke or generate an immune response by receptors called antibodies, which are in our body. Now antigens are usually proteins or they're polysaccharides, and they bind complementarily to the antibodies to cause the immune response. So there were all kinds of ideas about the nature of this response, but in 1955, Niels Jerne, a Danish scientist, set several things straight. Namely, he proposed that antibodies were already around in your body. They were present before the antigen came, and they combined in various ways that would enable some of them to bind many different possible antigens. Also, he suggested that when any one of these combinations successfully bound an antigen, or when it worked, whenever something was introduced into your body and it found an antibody to bind to it, that these antibodies would be ramped up in replication so that whatever is called for is amplified. Soon after that, in 1957, Macfarlane-Burnet expanded these ideas into the official clonal selection theory, which has 5 features that we're going to talk about today. So my only source is our textbook. It is Karp's Cell and Molecular Biology, Concepts and Experiments by Jeanette Iwasa and Wallace Marshall, and you can actually follow along with me in pages 666 to 668. So I have recreated figure 17.7, and we're going to go through the 5 steps just like we have them in the book. So you're going to have to I'm apologizing. You're going to have to bear with me for my artistry. 1, my markers are running out, and 2, I'm not a very good artist in the first place. But here we go. So step 1, each B cell becomes committed to produce one species of antibody. And you can see this in the top left here. We're going to start with a stem cell. Those stem cells are going to differentiate. We know about pluripotency and such, but eventually they're going to get all the way down to specific B cells. Now these specific B cells are going to start producing antibodies on their outside. And you can see that these B cells each have different kinds of antibodies that they make. So each B cell in our body is going to produce mostly the same. There are some duplicates, but really not very many. Only after their response is needed are they duplicated. So we're going to first diversify. You can see all the different colors of antigens. And we're going to talk about this 5th step down here at the end. We'll come back to it at step 5. But step 2 is that B cells are going to become committed to antibody formation even in the absence of an actual antigen. Now this is not shown super well by the figure, but I tried to delineate it here. We have an example microbe here, but this microbe is not actually going to bind to the B cells until later on. This is just to show that this section of the capsule, which is so conveniently blown up here as a little line with these dashes, it is going to bind complementarily to the B cells, but it doesn't quite happen yet. They showed this here, but it's not happening yet. It's just to show that that's what these antibodies are for. So step 2, we've bound we are committed to a single antibody formation, even in the absence of an antigen. Okay, step 3, we're going to make what are called plasma cells. Now, plasma cells produce many antibodies, and they don't keep them just in their membranes. They even secrete them into the liquid. So these B cells, which are immature B cells until they bind something, when they bind something, they're going to differentiate into plasma cells, and those plasma cells are going to produce a lot of antibodies into the serum. Now, we're hearing a lot about antibodies these days because we've got this virus thing going around. You probably heard about it. And we're doing these antibody tests to see if somebody's experienced that virus before. Now, the reason that this works is if they've experienced the virus before, their cells are going to come over here and differentiate it. And one of those differentiations is going to be able to bind something about the coronavirus. And that one is going to duplicate and make a bunch of these plasma cells which secrete these antibodies into your serum. And so they're testing for these antibodies here. Whenever they do those tests that are not happening in Texas and all this other stuff, not enough at least. Anyway, that's another subject. The other route that they can go is they can become mature memory B cells, B lymphocytes. And that's step 4. The immunologic memory provides long-term immunity. And the reason that this is important is the plasma cells secreting this into the blood, this peak secretion is going to drop eventually. And over your lifetime, you're going to need these memory cells if you experience it again in the future. Now, these memory cells are really going to boost and kickstart your immune response later, and they can basically come back here and do the same thing. They can make more plasma cells at a later time when they're needed, but we need some of these to stick around and memorize what they've faced before. and there will be several of these around. So the fifth step is sometimes the most important. We've all heard about immune disorders. And immune disorders are what happens when your immune system accidentally attacks you instead of just the actual pathogens in your environment. So you can see that this cell right here, when we were all differentiating over here, each one of these made a different antibody, but this cell made green antibodies. Now in my figure, I'm going to say that green, just like the cell membrane, is going to mean an antibody that goes against self. Now this is not what we want. This is very bad. So we're going to take this guy over here, and it's going to induce apoptosis, okay, before it ever has a chance to attack. Most of this is done
in the lymph nodes and in the bone marrow
while it's maturing. And most of the time, healthy bodies will get rid of any kind of self-antigens. So that means that the immunologic tolerance prevents the production of antibodies against self. And that's very key because we don't want to be eating ourselves alive with our own immune system. It's very bad for your health. So that's my presentation. I hope you've enjoyed. Here we are. So yeah, B lymphocytes, they're great. Immune system, it's good. Keep it up. Don't get coronavirus. And I'll see y'all hopefully later on. Enjoy.